Mammal DNA, too, proved eminently clonable. Well, this is not terribly surprising in retrospect. A piece of DNA, after all, is finally still DNA, its chemical properties the same irrespective of its source. It was soon clear that Cohen and Boyer's protocols for cloning fragments of plasmid DNA would work just fine with DNA from any and every creature. Phase 2 of the molecular biology revolution was thus underway. In Phase 1, we aimed to describe how DNA works in the cell. Now, with recombinant DNA, we had the tools to intervene, to manipulate DNA. The term recombinant DNA may present a little confusion in light of our encounter with recombination in the context of classical genetics. In Mendelian genetics, recombination involved the breaking and reforming of chromosomes with the result of a mixing and matching of chromosomal segments. In the molecular version, mixing and matching occurs on a much smaller scale, recombining two stretches of DNA into a single composite molecule. The stage was set for rapid progress as we spied the chance to play God. It was intoxicating, the extraordinary potential for delving deep into the mysteries of life, and the opportunities for making real progress in the fight against diseases like cancer. But while Cohen and Boyer may indeed have opened our eyes to extraordinary scientific vistas, had they also opened a Pandora's box? Were there undiscovered perils in molecular cloning? Should we go on cheerfully inserting pieces of human DNA into E. coli, a species predominant in the microbial jungle in our guts? What if the altered forms should find their way into our bodies? In short, could we, in good conscience, simply turn a deaf ear to the cry of the alarmists that we were creating bacterial Frankensteins? In 1961, a monkey virus called SV40, SV stands for simian virus, was isolated from rhesus monkey kidneys being used for the preparation of polio vaccine. Although the virus was believed to have no effect on the monkeys in which it naturally occurs, experiments soon showed that it could cause cancer in rodents, and under certain laboratory conditions, even in human cells. Because the polio vaccination program had, since its inception in 1955, infected millions of American children with the virus, this discovery was alarming indeed. Had the polio prevention program inadvertently condemned a generation to cancer? Well, the answer, fortunately, seems to be no. No epidemic of cancer has resulted, and SV40 seems to be no more pernicious in living humans than it is in monkeys. Nevertheless, even as SV40 was becoming a fixture in molecular biology laboratories, there remained doubts about its safety. I was particularly concerned since I was by this time head of the Cold Spring Harbor Laboratory, where growing ranks of young scientists were working with SV40 to probe the genetic basis of cancer. Meanwhile, at Stanford University Medical School, Paul Berg was more excited by the promise than by the dangers of SV40. He foresaw the possibility of using the virus to introduce pieces of DNA, foreign genes, into mammalian cells. The virus would work as a molecular delivery system in mammals, just as plasmids had been put to work in bacteria by Stanley Cohen. But whereas Cohen used bacteria essentially as copy machines, which could amplify up a particular piece of DNA, Berg saw in SV40 a means to introduce corrective genes into the victims of genetic disease. Berg was ahead of his time. He aspired to carry out what today is called gene therapy, introducing new genetic material into a living person to compensate for inherited genetic flaws. Berg had come to Stanford as a junior professor in 1959, as part of the package deal that also brought the more eminent Arthur Kornberg there from Washington University in St. Louis.
This book is continued on Disc 4.